So we've looked at these different kinds of time, interval timing, ordinal timing, and circadian timing. And the question arises as to what role, if any, does the hippocampus play in these different kinds of timing? And the reason for wondering about this is that, of course, HM was impaired in far more than just his ability to localize himself in space. He had a loss of episodic memory. And episodes are things that unfold over time, so they're not just a discrete event in space, but they also they play out in time, and so the hippocampus might plausibly be involved in that aspect of memory. So we're going to look at experiments that have tried to link the hippocampus to these three different kinds of time. So it's a large literature, and I've not got scope to go into it in detail, but I just want to give you a flavour of the findings that have linked hippocampus to some kinds of timing, but not all. So one of the early studies was a study of whether the hippocampus is necessary for interval timing, so for the timing of durations. And this is the experiment that we met in the last lecture, where rats had to discriminate stimuli based on either their duration or their rate. So if you remember that experiment, some of the stimuli were long duration and some were short, and some were high frequency and some were low. And rats could learn either of those aspects depending on how they were rewarded. So the question is the hippocampus needed for them to do this? So what they did was to transect the fornix, which is the structure shown in red, which is the fibre bundle that connects the subcortical inputs to the hippocampus. So structures like the medial septum and some of these important subcortical structures that mediate things like theta rhythm and cholinergic drive and aspects of emotion and other kind of very fundamental inputs. So if you transect the fornix, you disrupt the hippocampus quite severely. And in the days before we knew how to make really precise lesions, chemical lesions, excitotoxic lesions, fornix transection was commonly used. So what they found was that fornix lesion rats actually were not impaired on either the duration or the rate version of this task. So they learned normally. And that seemed to suggest that the hippocampus is not necessary for encoding the passage of time. However, there were a number of other experiments that seemed to suggest something slightly different, and this is experiments looking at the ability of animals to hold on to a memory across a delay. So this is an example. It was quite an influential experiment using a paradigm called the nictitating membrane classical conditioning paradigm. And this has been used for a long time to study associative learning, and it's done in rabbits because rabbits have this reflex called the nictitating membrane reflex. So if you puff a puff of air onto the cornea of a rabbit's eye, it'll blink. And if you warn the rabbit that this is going to happen with a conditioned stimulus, like a tone or something that's reliably precedes the air puff by a fixed amount, the rabbit will learn that duration and it will come to anticipate the air puff, and it will blink just ahead of the puff. So you can see the trace on the bottom there is showing you the nictitating membrane response. That's the membrane that covers the eye. And you can see that that response has come to just precede the delivery of the unconditioned stimulus. So this is a way of seeing whether the rabbit's brain is tracking time. So in the trace version of this task, there's a temporal delay between the presentation of the conditioned stimulus and the presentation of the air puff. And that trace interval requires that the animal hold on to the memory of the CS and track the time so that it knows when the US is going to appear. And it's compared against a delay version of the task where there is contiguity between the conditioned stimulus and the unconditioned stimulus. So there's no gap. And then there's a third condition where there's no relationship at all between the conditioned and the unconditioned stimuli. So in this experiment, they compared hippocampal lesioned animals with rats that had lesions to cortex, including retrosplenial cortex, and unoperated control rats. So you can see that the unoperated control rats learned the task quite well. So you can see the rate at which they anticipate, they blink in an anticipatory way, is increasing over the days of the conditioning. And similarly with the rats with the general neocortical lesions, but the rats with the hippocampal lesions or the retrosplenial lesions, so these are both structures implicated in episodic memory, they were both impaired and they did not learn the delay version of that the trace version of that task, which is on the left. But they could learn the delay version, which is the traces on the right. So they were unable to bridge that gap. And there's now quite a body of evidence showing that the hippocampus is necessary for spanning intervals, holding on to things across time. So whenever there's a delay in a task, that task tends to become hippocampal dependent. Whereas when there's no delay, then even animals with hippocampal lesions can do these tasks. So we still don't fully know the reason for this. It's clearly not to do with the timing aspect, because we've just seen that rats with fornix lesions could learn rate and duration. So there's something to do with holding on to the memory of the stimulus over that time. Now, ordinal timing, so learning the sequence of events, seems pretty clearly now to depend on the hippocampus. And the experiment that's been most influential in illustrating this is this experiment by Fortin et al. In the Eichenbaum lab, which we met in the previous lecture, where rats were presented with a sequence of odours, and then they had to choose between pairs of odours, and they had to select the earlier one of the pair. And we saw that normal rats could do this quite well, and those are shown in the pink bars there. But rats with hippocampal lesions could not do that. So they were unable to learn the ordinal relationship between the stimuli, although they could detect the stimuli, they recognised the stimuli. They knew they'd seen them before, but they didn't know when in the sequence they had seen them. And then the final kind of timing that we've met is circadian timing, and we now know that the structure in the brain that's responsible for circadian timing is not the hippocampus, but the suprachiasmatic nucleus in the hypothalamus. And I don't want to go into this in detail, but lesions to the SCN impair the ability of animals to show that cyclic 24-hour clocking of their activity and of other things that we saw earlier. So this different kind of timing depends on a different part of the brain. So of those three kinds of timing, the hippocampus seems to be important for learning ordinal, so the sequences of events, but not for learning specific durations and not for following the 24-hour day-night cycle. So in the next lecture, we're going to look at the cells in the hippocampus that seem to be involved in these aspects of timing that we've seen that the hippocampus is involved in.